Okay, so let's have a real lecture. Let's first of all introduce you what we're going to talk about in the whole class. So we get AEM 341. It's called General Microbiology. So we have 4 exams. What do we talk about? Exam 1, basic microbiology. Including numeration, identification, isolation, bacterial media, a growth curve, all kinds of the basic knowledge of the microbiology. Exam 2, we will talk about human pathogen. So for example, Haemophilus influenza, Neisseria gonorrhea, Streptococcus pneumoniae, all those major human pathogens in the United States we will introduce to you. So that means in this session of the exam we may have some case study, but we will talk about this detail in the lecture. Exam 3, this is usually relatively difficult. It's a combined with biochemistry and the genetics. So for example, we'll talk about the glycolysis, we'll talk about TCA cycle, but we're not going to talk about all those chemistry like biochemistry class teaching. We talk about is how the bacteria got ATP, the energy calculation. Genetics basically following the central dogma. So we talk about DNA replication, transcription, and translation. And that's exam 3. Exam 4, what we talk about is applied microbiology. Applied microbiology, typically we talk about antimicrobials, we talk about poultry meat processing, we talk about immunology. We also going to cover some lectures about like guest lectures. Okay? So, what all these says is the first slides, what we talk about. Based on the content for the exam 1, 2, 3, and 4, what is microbiology? That means science of microorganism. That's all we're going to talk about. So if you read the slides, microbiology is concerned with the study of microorganisms, including algae, bacteria, virus, protozoa, fungi. Okay, now let's talk about this. Go to the detail. First question. Since we mentioned the microbiology is a science of microorganisms, so we need to get to know the overview picture. What is microorganism? So this word is very good. It's very interesting. Microorganism. When we talk about this terminology for microorganism, we basically cut down here. We separate into micro and organism. So what means micro? It's too small. That's why we say it's too small, or we say too tiny to see by naked eyes. Therefore, in order to do the observation of microorganism, we need a tool. The tool called microscope. This is our first lab section next week. We'll talk about how we use microscope to do the observation of a bacteria in embedded slides. Now, obviously, microscope, there's a lot of microscope. Or we say microscopy. We have a light microscopy, we have a laser confocal microscopy, We have a bright field, a dark field, and electron microscopy there are lots of. So we are going to introduce in the lecture. But we will practice in the lab is using light microscope. Okay, so that's something. So micro, too small to see, by naked eye need to use microscope. It's a tool. So what is our organism? That's live body. Which means it's alive, some formation of the life. Okay? So then we go to talk about. Then what are the examples? You can look at the slides, it's complicated. Cellular, acellular, listed all these things, but I will introduce you in a little bit easier. So, we know every single live body can be categorized as a cell unit or without a cell unit. So that means we have cellular and acellular. Okay, then let's talk about what is cellular. When we talk about microorganisms, regardless of your background, you absolutely know bacteria. You heard about when you were very young. Maybe the first time introduced to you is just bad bugs. But the bacteria you know. And everybody knows one bacteria for sure is E. coli. Regardless of what your background. Even you study English or political politics, you know what is E. coli. But E. coli is a very simple name for one type of the microorganism, one type of the bacteria. There are lots of the knowledge behind that. It's a little bit tricky and complicated, but we'll get to that later on. So another example, Salmonella, you heard about. When you look at the news, every time you see Listeria. So those are some examples of the bacteria. Okay, acellular, let's jump to here. There's one thing for acellular, everybody knows. It's a virus. Why everybody knows? We have been suffering the last couple of years for coronavirus. Is that right? So you heard about the virus. Remember the time we had the masks, we have to stop the school and go everything online? It looks terrible. But you know the virus. That's acellular. Okay, what's a virus looks like? The structure of the virus is very simple. Like that. Okay, the outside, protein. Inside, genetic information. It is DNA or RNA, but never both. So then what we talk about, the really bad bug, like this, like that. We put a coating. This is like a crown. Okay, then what we get? We get a coronavirus COVID-19. That's what it is. Okay, these are the two things you know. So, but in a different category, let's talk about this. Did you ever heard about fungi? You heard about fungi. Based on their surviving environments, fungi have two different categories, which is yeast and molds. This is usually 37 degrees Celsius, this is 25 degrees Celsius at room temperature. So what is molds? You eat a bread for the breakfast. So you forgot about it. You left on your bench in the dorm, you go to a classroom. So you didn't clean it up. You left in the bench on your desk about a week. Then one day you woke up, you see the bread turns greenish color. So what's that? It's molds. What is yeast? Did you like beer? You drink beer sometimes. We all like beer, we drink all the time. Where they come from? It comes from the yeast budding fermentation process called Saccharomyces cerevisiae. So that's the yeast. Okay, so that's fungi, the second one. Okay, what else we have? We talk about the bacteria. We think bacteria is relatively well developed. There is some other type of the bacteria is not well developed, but it's still existing. We call archaea. So that's why there is a category here is archaea. Okay. And they gave you example is messaging. Archaea basically is similar to bacteria, but the structure are very unique. So for example, bacteria when they're moving, they have a special structure called the flagella. And the archaea does not have a flagella. They have endoflagella. It's very unique. But it's give our big contribution to the environment. So messages, for example, they developed a message. It is important. Remember? So talk about this. What's the angle? 109.4. Is that right? And then in chemistry talk sp3 hybridization, blah blah, all those things. That is organic chemistry. But we don't talk about this here. We just want to mention the messagens is one example of the archaea could generate the message. Okay. What else we have? Protists. So when you read, when you study, you know, we know bacteria, we know fungi. And then we know there is an old formation of the bacteria called archaea. Then what do we left? So you already know those three. Then we only need to study one more. Okay, then look at that. Protists. Then you look at the arrow there, you see there are different things. Protozoa. This is, you know, what is protozoa. For example, Toxoplasma gondii. If you like the cat, that will be the host. The intermediate host for Toxoplasma gondii is a cat. That's why the pregnant woman is not recommended to have a cat at home. That's protozoa. Then what else we have? Algae. This is usually looks like a plant. The plants is in the river or aquatic system. They are very unique. So for example, bacteria is using glucose, going through glycolysis to get energy. And the algae, what they do, they use photon or light as energy. So we call it a photosynthetic. And it's created 80% of the oxygen in the world. Okay, now which one we left? We haven't talked about is slime modes. Okay, this one you never heard, I never heard. What is slime modes? Slime and modes, which means two different phases during their growth status. They could be like a modes, they could be like a protozoa. What they do, if you have a debris or dusts, this guy usually can engulf and destroy and eat it. So this is a cleaning machine for the environment. It's very unique. It's belonging to the group which is protists. So that's what we learned for the cellular category of 4 different examples of microorganisms. Now we go ahead and talk about acellular. There is some easiest way to do, to learn. We mentioned about virus. We have a protein, as a coat. In the middle is DNA or RNA. We call it genetic information. So, we have a different category. This alone could be a microorganism. If this is RNA, that could be a very special microorganism called varroids. In 1996, in Philippine area, they have a big damage of those palm trees, which is coming from a plant pathogen. And the people later on find it is the only single RNA sequence. It is a varroids, we call it. It also could be the middle is RNA, and outside it could be packaged with type of some very simple protein. We call it a satellite. But I never see anything. I never touch satellite. I don't know about that too much. But just to let you know, one of the form of an acellular microorganism could be satellite. The last one. So we talk about the middle, RNA or DNA as a genetic information of a virus. How about this outside guy? The protein alone can be a microorganism. What is that? Prions, that is called. You may not know prions, but you heard about medical disease. This happened in 1997 in England, in Great Britain. The cow has been contaminated with a very special microorganism called prions. And they basically is a damage of their cells membrane system of the brain system. And it's very contagious. The only way they can do is killing and burning and also do the deep burial. And they transferred to the United States, unfortunately, in 2003. It gives a big damage for the meat industry, especially beef industry in the United States. So even today, Australia, Russia, and China will be forbidden if we ship any of the beef products to there. So for example, if I want to ship one bags of beef jerky to China, it will not pass the customs board because based on the regulation, they may think the beef jerky will be contaminated with medical disease. So that simple protein, prions, is in the history cause a big damage for the beef industry in the United States. It's related to the medical disease. Okay, so this is what we gave you introduction of these slides of the table, the figure where we talk about the microorganism and the examples. So the next question for you is very simple. Do you think how many microorganisms are in the world? It's huge. How huge it is? It is more than 10 to the power of 30. It is huge. And most of those are less than 1 micrometer size. Our gut area is a big accumulation of microorganisms. People think it is a central of our body. Our environment has all kinds of microorganisms. Some things are very special. So for example, if you go to Yellowstone National Park, in that very hot springs, you see the bacteria still there. Those are highly resistant to the heat. If you go Salt Lake City in Utah, you see the big salt lake have lots of the bacteria. Those bacteria love the salt. We call it halophiles. So just want to let you know there are lots of millions, trillions, or people make a joke that gazillions of the microorganisms. But how many we know? less than 5%. That's the reason when this guy happens, people is not prepared. Even we did so many work for the virus, because think about that. There is 100% of the bacteria. We only know 5 microorganisms. We only know 5% of them. Something happens, we are not prepared, but we learned a lot during this pandemic. So develop the vaccine, develop biotechnology, we learned from that incidents. Okay? So this is something we want to let you know is the overview of the microorganism. We get a very limited time for the lecture. So in this class, we basically will focus on bacteria. And in the lab, we will using is biosafety level 1 bacteria, which means it will not cause a severe risk for the human health. We will learn a little bit about modes in one section of the lab. And I will prepare a very molded bread. And we use a very simple method to isolation. And underneath the microscope, we can see the morphology of the modes. Later on, maybe in the early November. We will not talk about the protozoa. We don't have a facility to talk about virus. So virus we'll mention very briefly. One lecture in our last section, which is one lecture in exam 4, we'll talk about virus. So most of the time, we are going to focus on the bacteria. Okay, so that's the goal of the class. Okay, next one. Classification of the microorganism. Microorganism, in the history, people using the genetic distance between the isolated ribosome RNA can categorize microorganisms into 3 major categories. Is bacteria. We call it true bacteria. I call it all the formation of the bacteria is archaea and eukarya. So bacteria, archaea, and eukarya are the same major classification of microorganisms. So the picture which is showing there, okay, this different category. Next one. How we understand the microorganism? Everybody knows bacteria is really bad. Microorganism is bad. That is our first impression. So for example, they will cause lots of the human disease. Let's say E. coli. Will cause diarrhea, urinary tract infection. We have a one lab section, we'll talk about urinary tract infection. We're testing our urine sample. Salmonella, it's caused food poisoning. It's a major pathogen in poultry meat. We'll have a special section talk about that. Virus, obviously, we already know. COVID-19, flu, hepatitis, and rabies, all those things. So give you an example of E. coli, I just mentioned. This is the pathogen, or the bacteria everybody knows. But there's a big story behind it. Most of the time, we talk about E. coli. Is the normal E. coli, which is a normal bacteria, is proliferate in our stool samples. We call it generic E. coli. So generic E. coli is used indication of fecal contamination. Second thing, if it is E. coli, 9037, the special species, this is used for fermentation. It's a beneficial bacteria. Why we are feeling terrible for E. coli? Because they are very special strain. E. coli 0157H7. We'll mention this in the exam 4. This is a pathogen, zero tolerance in beef products in the United States. Caused a big problem in the history. It is almost because of that we had our defense system, like HACCP, like sanitation standard operation procedures, like FSMA even, Food Safety Modernization Act, all started from that passage. Urological E. coli, which is specifically coming from urinary tract infection, but it's not only stays there, it's already spread from our food supply systems. So just want to let you know, even a E. coli, there's a big story behind it there. There are lots of the research we can do, not only stay on saying this is a general normal bacteria. Okay, bacteria also bad. Some of the examples. The fish could have bacterial infection caused by Vibrio. The tomato could be contaminated with bacteria. You see a very lotted blossom caused by a plant pathogen called Aeonium. You see the human being's eyes is conjunctiveness of eye. This is really terrible. If one day you have a conjunctiveness of the eye, that does not mean it's a skin contamination. You may have a big trouble because it's a very good indication for sexual transmitted disease. Let's say chlamydia. Let's say we talk about chlamydia is one of that, or we have other related sexual transmitted pathogen could cause conjunctivitis. So that you need to look at your normal life, what it looks like. Last one, strep throat. This is everybody knows, everybody heard about that. And I can tell you something. We require 2 attendance for the lab. Then start from November or later October, people are going to send me emails saying I'm not going to come in. Why? Because I tested positive of strep throat. That is one of the excuses before COVID-19 happens. And we will learn this in the classroom. What is strep throat? That's what it looks like. It's caused by Streptococcus pyogenes. We also call it a group A strep. It's a very invasive pathogen, but you can see the tonsil area have inflammation, very obvious. So we talk about the bad things for the microorganism. We talk about good things. What are the good things? There are the beneficial things for the microorganism. We just mentioned earlier, everybody likes alcoholic beverages. You look at the beer, you like it. The 2 types of the beer, the ale and the lager. So top and the bottom fermentation coming from the yeast. So yeast fermentation. The wine, look at the wine, you like it. Where they come from? It comes from the grape. It's a grape fermentation. You have a red wine and a white wine. Antibiotics. You heard about penicillin. Antibiotics usually is a metabolic products coming from one microorganism and killing or inhibit another microorganism. So antibiotics. All the time, antibiotics you cannot orally take. You have to give a shot at your butts. Okay, so that's all time. But right now, lots of the products is orally taken. Insecticides can kill insects. Other things, everybody likes those fermented products. Cheese, Swiss cheese, cheddar cheese, all kinds of cheese. Yogurt, and those sour cream. Which bacteria cause these type of products? Going through the fermentation is lactic acid bacteria. It's very simple, lactobacillus. This we will talk about in exam 3. When we talk about the fermentation at the biochemistry section, a little bit about the chemistry there. But we will let you feeling comfortable to learn about it. So that's a good thing for microorganisms. Okay, after I talk these slides, we'll be off. I'll let you off a little earlier today because it's the first lecture. When we give you some example of the microorganisms and talk about the bad things and the good things for the microorganism, then why microbiology is developed so well as a science in these days? Because our ancestors or we say those early scientists laid down a good foundation for us. A very easy example is a vaccination. So let's talk about that. I usually talk about that earlier. The reason is, by the time, last couple of years when COVID-19 happens, we always have a debate about the vaccine. If you look at the news, they're debating all the time. So that's why I have these slides earlier, to let you have a very brief information about what is vaccination. And the detail we will talk in the exam 4, applied microbiology, when we talk about the immunology. So, vaccination. So this terminology is very interesting. What is vaccination? This is coming from VACCI. What this means? In Latin words, this means cow. So that means there is some story behind the cow. So what's that? Edward Jenner, a medical physician at Royal Community in Great Britain, back in somehow like 17th century, he wants to do some of the research to solve the problem of smallpox. So smallpox caused millions of people died every year in European countries. It will be damage the immune system, cause immune deficiency. Edward Jenner find it very interesting. There are one group of people are naturally resistant to smallpox, which is a woman working in the dairy farms. And he find that the woman working in the dairy farms, they will not get smallpox. Instead, they will get a very mild symptom called cowpox. So what he did, he get the arm from some of the cowpox patients coming from this Ling area, get the liquid from that, do a very simple purification, and get the liquid, and then to inoculate or inject this baby from the royal community in the Great Britain, and then contaminated this baby with smallpox, and this baby survived. This is the first time an example for vaccination. And later on, people want to memorize Edward Jenner. That's why they come out this terminology called vaccination. And the VACCI means cow. So what is vaccination? Using a tiny dose of the toxin injected into your body to stimulate a mild immune reaction in your body. It could generate antibody then against the foreigners. At this moment, it is smallpox. Okay, so Edward Jenner is the first time they established the role of microorganisms in a disease. And I always say that, in the textbook also say that, for the virus, including coronavirus, smallpox, if it happens, you do medical treatment is always challenged. The best way to conquer the virus is the vaccine, is the vaccination. So we will show you in the exam 4 when we talk about. In the United States, the newborn baby have a very strict immunization schedule from their newborn until they are grown up to the adult. So for example, you get a couple of shots like DTaP, diphtheria, tetanus, pertussis. You get a couple of shots with streptococcus pneumoniae called the PCV, or you get a couple of shots with HIV, which means type B Haemophilus influenza. So just want to let you know the best way to control or to prevent the virus disease is vaccination. Okay, so I will end up today. Next Tuesday we'll start talk about discovery of microorganisms.